Hello, everyone, and I would like to welcome you all to this seminar series, and I'm very delighted to introduce you to today's speaker, who is Yasemin Hocam. And a brief about herself. Yasemin Hocam, she got her bachelor degree from psychology department at Hacettepe University in Ankara in 2006. And then she went to the United States, American Texas University in Dallas, where she studies the applied cognitive and neuroscience for her master's studies. And then she went to, she stayed in Texas again for her PhD studies. And she was at Texas University and Southwestern Medical School. Again, she studied neuroscience. And then in 2018, she went to Boston, where I met with her, actually. She was serving as postdoc at a Tufts University. And then she completed her postdoc studies, and then we convinced her to come to Kadir University. It was the first time we met in Boston at MIT Lounge, right? And she was among, and Deniz Hoca, we also convinced Deniz Hoca at Northwestern University, and then we had another stop in Boston, where I met with her, and I'm very delighted that she became part of Kadir University. Of course, Deniz Hocam became also part of Kadir University, and she will talk about biological clocks, neural circuits, and metabolism symphony. And the floor is yours, my dear Yasemin Hocam.
Thank you. Can you hear me well, by the way? Okay. Thank you, Sondan Hocam, for this very kind introduction. And I would like to say, actually, you really did convince me. And I remember being very inspired by your presentation that you gave at MIT that day. And that really motivated me to come to Turkey. And I'm very happy to be here at Kadir Has University. So today, I'm going to tell you about biological clocks, neural circuits, and the Metabolism Symphony. And Nihat Hoca actually cannot make it today, unfortunately, but this symphony was his addition to my title. He said it's going to sound more sexy, so we put it that way. And I really liked it. And I actually really like this orchestra analogy, and I'm going to use this a little bit. Oh, also, before I start, I would like to apologize for not giving this talk in Turkish, but I have a good excuse. I have international students. They are here, actually. So I wanted them to be able to follow this as well. In the future, hopefully, I will give a talk in Turkish as well. Back to the biology. And also, I'm going to talk about a number of biology concepts today. If you have any questions, please feel free to ask me along the way. I like this orchestra analogy, and I'm going to use this a little bit, as I said. So in an orchestra, rhythm and synchrony is important. So is there anybody who likes classical music here? Raise your hands. A lot of people. So you've been to an orchestra concert, right? There is an orchestra and there's a conductor. When you go to a concert, you know, you have all these expert musicians playing their instruments. You have the violin, you have the trombone, you have the viola. These are all experts, talented musicians. And you may think, you know, what is this conductor doing here? Like all these crazy movements. What is the significance of it? And actually, what they do is very significant, because if the orchestra is out of tune, all the magic goes away. And I want to show you a little video, if I can, hopefully. Just a little fun video that I hope I can show really quick. And I think it's good because it's New Year's is approaching, so I can celebrate everybody's New Year. Just wait for it, it's coming. So Happy New Year, everybody. So it doesn't sound great, does it? And I think this is a good analogy. So now I'm going to come back to biology and the metabolic homeostasis. Actually, this you can think of it as an orchestra, and the orchestra conductor in this case is the brain. But you have all these organs, metabolic organs, with important functions. You have the liver, pancreas, skeletal muscle, white adipose, brain adipose, and you have a constant back and forth between these organs. So the brain is taking signals from the environment, such as differences in temperature, the light information. It gives information about the time of the day through the retina coming from the eye to the brain. And also the brain takes the hormonal and nutritional signals from the circulation, and then it responds to these signals either by the autonomic nervous system signaling back to these organs. It also directly affects the food intake behavior. So what makes us eat versus not eat is this hypothalamus region in our brain. So, and in my talk, actually in the first part of my talk, I'm going to tell you about the control of, about peripheral control of metabolism. I'm going to tell you a little about the brown fat and a study that I did during my PhD studies at UT Southwestern, where we discovered a role for a circadian protein in cold adaptation in brown adipose tissue. And then in the second part of my talk, I'm going to come back to the brain, and I'm going to talk about the central control of metabolism and how the neurons in the hypothalamus region of the brain communicate to each other, and how this communication changes by different metabolic states or feeding states. And again, coming back to the orchestra, it's a good analogy, just as the metabolic state, the orchestra conductor, they would always steer the orchestra, not to the audience, so they can follow what these instruments are doing, and they can signal to them, do all those things, and the instruments, musicians will respond to this conductor, and that's how you hear this beautiful, harmonious music. And what happens if this balance is disrupted? We would have all these metabolic disorders if this homeostasis, this metabolic balance is disrupted, such as obesity. And we know obesity is a major global health challenge, both locally and in our country as well. In this map, this is estimates by World Health Organization 2016. This is predictions of estimates of prevalence in obesity, and you can see that one of the highest prediction is our country is among the 30% and more prevalence category. And why do we care about obesity? There's a lot of obesity-related comorbidities such as heart disease, diabetes, stroke, hypertension, cancer, and in the past 3 years we've been through this pandemic, COVID-19, and being obese was also a risk factor for having more severe onset of symptoms for COVID. And in order to be able to understand these diseases, we really need to understand the circuitry behind it and the molecular mechanisms behind it. And it's rather complex because there's so many components of it. It's a coordination between the brain and all these peripheral organs. And also, our bodies have a rhythm. So here you see a lot of the physiological processes in our body have a rhythm, and this is simply to adjust the physiology to the day-night cycle of the Earth, of the 24-hour rotation of the Earth. So the organism wants to keep all its function at the most efficient level. So it doesn't want to waste any energy needlessly. And when there's a lot of food, it wants to make the most use of it. So this is why actually there are different metabolic processes that's active during the wake cycle, which is expected that you will be eating, such as actually you do a lot of synthesis at that stage, like glycogen, cholesterol, and bile acid synthesis. You secrete a lot of insulin. You actually produce fat because the body is expecting a lot of food coming in. So it wants to turn it to something else so that it can store it. And glycolytic metabolism in the muscle. And when you're asleep and the body expects you to be fasting, other mechanisms such as gluconeogenesis, glycogenolysis, glucagon, lipid catabolism, leptin secretion, oxidative metabolism. So actually the body is conditioned to burn the fat because it's expecting for you to not be eating. So actually this is the best time for you to burn fat. If you want to lose weight, this is why you shouldn't be eating at night, because when you eat at night, you convince your body then that it switches it to the wake state, and now it wants to make fat again. So circadian biology is important. The timing is important. So for this, everything to occur in a balanced manner, everything has to happen at the exact time. Exact certain time. And the circadian comes from Latin circadium, meaning about a day. And also this rhythm also occurs at a molecular level. So at a very simplified way, you know, all our, this is the core of the molecular biology. We have the central dogma. We have DNA in all of our cells. This DNA is transcribed into RNA and then translated into protein. And the DNA is this compartment, this executive compartment, which would have all this information. And actually, RNA and protein is what goes and does things in a cell. These are more functional. So if I were to look at all the DNA in all of your cells, DNA would be the same in all your cells. But the gene expression, the RNA levels would be very different from on like a neuron cell versus a liver cell. Because what makes the differences in a cell is due to gene expression, not to the DNA content in the same person. And even if I look at two different neurons from the same person, the gene expression would still be different. So these are the functional compartments that we really look to understand, to take a snapshot of the cell of the organism in that exact moment to see what's happening in the organism or in the cell. And we talked about this rhythm. We talked about there's the light and dark phases, and the organisms adjust their physiology accordingly to this daylight cycles of the earth. And there is a behavioral rhythm, you know, at night you will be asleep, you won't be eating, hopefully. And during the day you are awake and you will be eating. And at the molecular level, so the circadian thing that I told you about is controlled at a molecular level actually, and in the past decades, the really molecular mechanism of it has been discovered. There have been a number of studies, so we now know how this happens. So there are actually some a core clock machinery, and there is this what we call positive and negative elements. So these are proteins that would bind to the regulatory region of the DNA, and that would induce the expression of a certain number of target genes in a rhythmic manner. So these would be rhythmically expressed genes, and approximately 10% of all expressed genes are rhythmic. And also I should mention in 2017, you might have heard the Nobel Prize in Physiology went to Jeffrey Hall, Michael Rosbash, and Michael V. Young for their discoveries in the molecular mechanisms controlling the circadian rhythm. And I could not mention Aziz Sancar. Actually, he was a source of pride for all of us. I was in the US when he received this, and this was actually very exciting for me, and it was very exciting for circadian field and in general, because this was actually, although Aziz Sancar got this Nobel for discovery of DNA repair mechanisms, actually, he also found that this DNA repair mechanism occurs in a circadian manner. And he also discovered one of the very important core clock molecules as well. So he was actually the first circadian-related Nobel awardee in that case. So after this little introduction, I'm going to tell you a little bit about my PhD work on with a circadian protein and how it regulates metabolic adaptation in brown adipose tissue. I did the studies at UT Southwestern Medical Center in Carla Green's lab. This Nocturnin protein, my PhD mentor Carla Green, she discovered this in Xenopus laevis in a rhythmic screen, in a high throughput screen for rhythmic genes in this African frog retina. And after it was discovered, so this was a gene that was under this circadian control. So its expression levels are rhythmic, and here you see this is from mouse tissue in the mouse liver and the mouse muscle. You can see the expression profile of this gene, and you can see that basically its expression peaks at the onset of nighttime, and please be advised that this is mouse tissue and mouse are nocturnal, so this is actually the active time for the mice. So it peaks just before the time that mouse becomes ready to eat, essentially. And also, in addition to being under circadian control, Nocturnin can also be acutely induced in response to a number of stimuli like LPS, serum shock, or nutrient status. And also, then further studies that Carla did, so they made this Nocturnin knockout mice. So when they removed this protein, this gene in mice from birds, this mice has an interesting metabolic phenotype that they remain lean on a high-fat diet. So, and they don't develop fatty livers, and they have this sort of metabolic benefit kind of condition. And so it was a curious case of what is the mechanism for Nocturnin to do this. Nobody knew at the time. And what the first hypothesis was Nocturnin actually works on some mRNAs, because it was thought to have a deadenylase function. So the idea was that Nocturnin will affect the stability of some mRNAs and thus affect metabolism, but we didn't know at the time. So when we look at the mRNA, here I'm showing the Nocturnin mRNA. So when we did a bioinformatic analysis and look at the sequence of the Nocturnin, so there is two potential translation initiation sites, and in between them there is a mitochondrial targeting sequence. Again, you can find this out with some prediction tools that's available freely online. So if this mitochondrial targeting sequence is functional, you would expect Nocturnin to have two isoforms actually. The protein, one isoform would have this mitochondrial targeting signal and would go to the mitochondria, and there will be one without this signal which would remain in the cytoplasm. So I did an experiment using some molecular biology to test this. So if this is really functional, and if we manipulate and force this transcription artificially to have this mitochondrial targeting signal or to not have it, do we really see what we expect to see? And basically, here you see some microscopy images. So here we are looking at the Nocturnin. This Nocturnin has a flag tag, and we put this tag because at the time we didn't have a really good antibody for Nocturnin. So this flag lets us visualize Nocturnin. And this is the full-length Nocturnin, so no manipulation done. And we see that Nocturnin is both in the, and the red stain stains for the mitochondria, by the way. So in the full-length Nocturnin is both in the cytoplasm and in the mitochondria. But when we force the Nocturnin to not have that mitochondrial targeting sequence, now we see that it's only in the cytoplasm. And when we force the Nocturnin to have that mitochondrial targeting signal, now we only see it in the mitochondria. So this told us that this targeting sequence is necessary and sufficient for Nocturnin to localize into the mitochondria. And after this, so we know that it goes to the mitochondria, but what does it do in the mitochondria? What is the functional significance of it? So basically I did a plate-based assay. This is done with cells. This is basically, these are all mouse embryonic fibroblast cells. So these are cells in culture, and you can do a mitochondrial respiration assay. Basically, you just plate your cells, you feed them into a machine, and this machine gives you a reading for oxygen consumption rate. And by applying different inhibitors for the different parts of the electron transfer chain in the mitochondria, you can measure different aspects of mitochondrial respiration. And without getting into too much detail, so this is just basal respiration without any manipulation. And we didn't see much of a difference here, but where we see a difference was this uncoupled respiration. And this uncoupled respiration is, again, without getting into too much detail, this gives us an idea about the maximal capacity of the cell for respiration. And this mimics an energetic challenge condition for the cell. So we are seeing a decrease in the Nocturnin knockouts in the uncoupled respiration. And actually, we were able to rescue this defect if we put Nocturnin back in. So far, all these experiments that I showed you, these were done in cell lines. But we really wanted to know what's the significance of this physiologically in vivo. So we wanted to look at a organism like mouse, and what is the tissue that uses this uncoupling respiration is brown adipose tissue. Brown adipose tissue basically use this for heat generation. So normally, this is how mitochondrial respiration happens, electron transport chain. So basically, there is a electrochemical gradient in the mitochondrial membrane, and you have all these protons pumped out, which would create this proton motive force, and this force literally turns this ATP synthase pump, and this is how you generate ATP normally. But if you uncouple this by means of, in the case of the brown fat, there is this UCP1 protein, which is an uncoupler because it would pump the protons back in. So you basically uncouple the mitochondria, and now all that energy would be lost as heat instead. And this is a mechanism for organisms to, actually one of the mechanisms that help organisms defend their body temperature. And we call this thermogenesis. And so coming back to nocturnal knockout mice, there was an interesting phenotype. So under standard diet, their temperature were slightly low, and under high-fat diets, this became even more significant. So did these mice have a thermogenesis defect, was what we asked. And so I started working on this brown fat now in mouse tissue. And first thing I looked was, do we really see nocturnin in the brown fat? So this is in the brown fat tissue from mice. I used a fractionation assay. So I isolate the mitochondrial and cytoplasmic compartment, and in both compartments I see that there is nocturnin indeed in the brown fat. And also, very interestingly, I also saw that upon a 4-hour cold exposure, so we just put the mice in cold temperature for 4 hours, and then we look at their gene expression. And I see in the brown fat tissue, and I see a robust induction of nocturnin after 4 hours in this mice brown fat tissue. And when I looked at thermogenesis, so now I'm putting the mice in a cold temperature, and I'm actually measuring their body temperature every 4 hours. And you can see that, so these mice do not have a total thermogenesis defect. They can still defend their body temperature. However, there is a slight delay which becomes apparent at the 8-hour time point after the onset of cold. So, so next, we wanted to look at the overall transcriptional response to cold exposure. So if nocturnin is doing anything to the mRNAs, we expect to see some differences in the overall transcriptional response. So this is the experiment we did. Again, we have the wild-type mice, nocturnin knockout mice. Our control group is kept at room temperature, and we have a cold exposure group. This first experiment is 4 hours. And then we collect the tissue, and we use this for RNA sequencing. And we also use the same tissue for metabolomics studies. And so we see some alterations in the transcriptional response, although the overall cold response genes, for the most part, seem to be activated in the knockouts. However, when we just look at the wild-type upregulated and downregulated genes that are specifically upregulated or downregulated in the wild-type, we see that there are more genes that is affected or altered specifically in the wild-type in comparison to the knockout animals. And then when we do a gene ontology analysis to see what these genes are related to, we see these are all genes related to some metabolic processes. And I analyzed some of these data myself, but we also took help from Stefano Berto and Konopka Lab for this bioinformatics part. So then what we thought was, we also wanted to look at metabolites because we are seeing these differences in the metabolic genes. So we did this metabolomics assay. Again, so this is an unbiased approach to see differences in metabolite levels. So here again, we are looking at 4-hour cold exposure, and we see that, so we are here, you can see the heat map here, you're seeing a PCA analysis, and you can see that the PCA analysis separates between the room temperature and cold conditions, but does not really separate between genotypes. So this shows that this metabolite profile is not that different between the genotypes. So then we thought that maybe 4 hours is not enough for us to see the metabolites, changes in the metabolites, because if this is, so nocturnin is induced at 4 hours, then it's going to be, at gene expression level, then it's going to be translated into protein, and then it's going to go and do some things on the metabolites. So we decided to do a longer cold exposure experiment. Here we are putting animals in the cold for 1 week and doing the same experiment basically. And when we look at that, we see quite a different profile now, where again in the PCA analysis now the wild type room temperature and cold and the knockout room temperature conditions are clustered more closely to each other, and the Nocturnin knockout seems to have a very distinct metabolic profile. So this was very interesting, and this was actually a great, very productive collaboration with Prashant Mishra and Bogdan Bordiani helped us with this metabolomics measurements. Yes. Sure. Sure, yes, please.
Okay, you know, while you were presenting these slides, right away the question came to my mind. The people who are living in polar region of the planet, who are living in cold weather, their metabolism are different from ours?
Could be.
Yeah.
Could be.
South, there's no one in South, but in North there is. And the Siberia, and so, their metabolism, because you did it on, but also.
And they are thinner than us.
Huh?
They are thinner than us.
Yeah, actually, if you go, well, I don't know if there's a, but
actually,
they
have clothes, and they're not
they have
cold.
Clothes,
actually,
they're not just But like, so that's a great question. It is very likely their brown fat is more activated because they are more exposed to cold. But you know, obviously they are being protected. They are wearing warm clothes and stuff. So it could be. But I should also say that like this brown fat is one way of temperature defense, but it's not the only way. You know, you also have. So this is called non-shivering thermogenesis, but you also have shivering thermogenesis that happens in the muscle. You know, you do that shivering, and you know, you also have the browning of white adipose tissue, which we're not gonna cover here. You also have some autonomic response like the vasodilation. The vasodilation versus constriction, so that you will have more blood flowing. And the temperature control actually happens in the hypothalamus as well. It's controlled by the hypothalamus as well. So this brown fat part is really a part of the story. And also another interesting point, in the humans actually, it was thought that there is no brown fat for a long time. But in the recent years, actually, they found out that we do have brown fats. And actually, there has been some attempts to find some drugs that activate this brown fat as like a like anti-obesity drug. People have worked on that, but some early attempts ended up very well because you can induce hypothermia, and people actually died for that. So it's a very again, it's a very intricate balance that you don't wanna. If you push it too much to one part, you can disrupt the whole balance of the metabolism. But coming back to your question, I think it's very likely that they have a different metabolism, and they are usually, I don't know. It's a good thing. Are they skinnier? Maybe they are skinnier. It's supposed to be. It's very possible. But this is only a tiny part of that temperature regulation story, but it's very possible.
Thank you.
Any other questions before we move on? Sure. Mikrofon kapalı galiba.
People living in cold areas, their mitochondria is specialized to produce more heat comparing to ATP. People living in hot areas like equator, their mitochondria specialized to produce more ATP comparing to the cold areas, which they produce more heat. that is why the people living in equator, they run better than the cold area, okay? Because when they run, they produce more heat. So that hinders the running, okay? So the people living in like Africa, they run better because their ATP, their mitochondria produces more ATP, okay? More ATP means more energy, okay? That gives you more effort to the muscles, okay?
Yeah, here you're stealing
That's
away
another,
from,
side of the research.
Thanks for that contribution, Kemal Hocam. Yeah, because for this mechanism, if this mechanism works a lot, you're stealing from that ATP production in expense to generate heat. So that's an interesting point. Okay, so now after we saw this, so this was an unbiased screen. We are looking at the metabolites as a whole. Then we saw this interesting distinct profile for the nocturnal knockout cold group. Then we went and looked at the now top metabolites at this principal component 1, which is the coefficient which has the most variance. And this was actually very striking. The top 10 metabolites actually with the most variance was TCA cycle metabolites. And TCA cycle, if you have any biology course at some point, you will remember this. This is the very important cycle that happens in the mitochondria, and all of these metabolites are used for oxidative metabolism, mitochondrial respiration. So, and we are seeing, and we didn't see this profile for the 4-day cold exposure group. So this was very striking to us. And then, so when we went and now look at these metabolites, that just graph them individually. So at the 4-hour cold exposure, both in the knockout and in the wild type, we see this induction of succinates. And in the 7-day cold group, this induction, this initial increase is blunted in the wild type, but the succinate levels remain strikingly high in the knockout animals. And a lot of other citric acid cycle intermediates also remain at high levels. So this suggested us, or how we interpreted this, was Nocturnin is basically helping to suppress this initial metabolic response in response to cold exposure. And when you don't have Nocturnin, so maybe in the wild types they're switching to some other metabolic mechanism, but when you remove Nocturnin, you are